But don't go beyond that. Like, nothing to know.
Okay, so I'm going to talk about genome-wide prediction in plant breeding, practicable or otherwise. I mean, it's a general topic, but so this would be the general plan for today's talk. Starting from Brassica natus, as Andre already told about Brassica natus, it's an amphidiplide of Brassica rapa and Brassica oleracea. So we have 38 chromosomes in Brassica natus. Now coming to my actual project, the aim of my project is to develop genomic prediction models, simply to identify best parental combinations for high heterosis. So the material I'm using is the diverse population of canola, rapeseed. We have two inbred lines, as Andre already said, crossed with 475 genetically diverse males. And we have 950 hybrids. These have been tested in different locations already in Germany. And the phenotypic data have been obtained by the company. And now we will use SNP data. We will use transcriptomic and metabolomic data also with the collaboration of IPK Gauss-Lieben in Germany. So these are some of the glimpses of my lab work the last 3 months. I mean, just some, but there are a lot more. So as you can see, I started from simple plants, and then I collected the leaves, and I went through different processes. But anyway, at the end, we got the DNA, and as you can see, this nice labels here. Okay, breeding can be classified in or put into two categories in general, or in two eras actually. We have a classical breeding, and we have a molecular marker era, which is post-molecular marker era. So that is marker-assisted selection and genomic selection. So what happens in classical breeding is that we take only phenotypic data, or field data, you can say, from individuals, their close relatives, or their progenies, and we predict the genotype of the next generation. Now, the main advantage of classical breeding is the tremendous increase in the yield of different crops and grains. And as you can see, there is a steady increase in the production of different grains in the last century or so. But there are some issues attached to such kind of methodology. The first one is expensive phenotyping. I mean, you have to do the field testing for each cross. And there's a large breeding cycle, so the time factor is also involved in that. And some favorable alleles of small effects are mostly ignored in the case of complex pairs. And it takes a long time to declare a variety, almost 15 to 20 years. So that's a classical breeding. Now we come to molecular breeding, which is marker-assisted selection. So it is used in plant breeding, but it addresses only genes with large effects, marker-assisted selection. So the question is, what about many loci for small effects? Here we have an example of milk fat percentage in dairy. So I borrowed this from Animal Sciences actually, just to give you a sense of the. So here we have this trait, which is a simple trait actually, which is controlled by DGAT1 gene. And we have an example of overall complex traits, which you can see is controlled by very many loci of small effects. Now coming to genomic selection. Mirvissen and his colleagues in 2001 produced this paper, which actually gave birth to this kind of methodology. They call it genomic selection. So what happens in this genomic selection, that we use all available markers, genome-wide markers, simultaneously to estimate the breeding values. And then we do selection based on those estimated breeding values or genomic estimated breeding values. So that was the general definition. Now, genomic selection can be executed in 4 basic steps. We have a training population where we have phenotypic and both genotypic data, as you can see here. And then we estimate marker effects in the training population. So we use both the phenotypic and genotypic data. And then we come to breeding population or validation population where we use both the marker effects and the genotypic data. So here we don't use the phenotypic data. We only use the marker effects. And here we use just the SNP data. And then we go for selection or genomic selection. Now the question is whether genomic selection is a practical way to execute. Well, in animal breeding, it has already established its values through different real data sets. In plant breeding, the phenomenon is still in its infancy, but there are still some studies which says that there's the highest accuracy of genomic estimated breeding values. And these are some of the papers which one can, if someone is interested, so they can read. Now coming to the advantages of genomic selection over the classical method. The first advantage is that it is a quick system because you have a low breeding cycle. So they call it a breeder's equation actually. So if you have a low breeding cycle and the accuracy is high, the accuracy of selection in genomic selection is also high. And then at the end, you select the top one at here. So it means you increase the intensity of selection. And this all means you increase the genetic gain or the genetic improvement. So it has many advantages actually. Now coming to the statistical model used in genomic selection generally, there are different statistical models used in this area. RBLUP, for example, Lasso, Bayesian Lasso, ORCA-HS, Bayes A, Bayes B, and so on really. I mean, there are quite a few actually. So I was really confused what model is to follow. And the question is, which model is to use? Now, we can generally put these models into 2 categories based on the prior assumption of the markers. For the BLUP, the assumption is that the effects of all markers are normally distributed with equal variance. And it is used in the case of polygenic traits. The assumption for the Bayesian statistics or models is that most loci have no effects at all. Only a subset of loci have a larger effects, and it is used in the case of oligogenic traits. Okay, finally coming to the conclusion of genomic selection. Genomic selection is becoming the most efficient way to improve complex traits using DNA markers, obviously, or SNP data. There is no single genomic selection model to be used as a benchmark in plant breeding. So the choice of the statistical model actually depends on the genetic makeup of the trait, population structure, and marker density. So the take-home message for today's presentation would be that cost-effective genomic information is reshaping the plant and animal breeding, both equal. The gap between the ever-increasing demand due to population explosion and the existing trajectory of yield could be minimized by embarking upon novel, efficient, and cost-effective or cheap breeding methodologies. Genomic selection could be a better option in that respect. So I would like to obviously thank my professor, Aarhus Denmark, and some people who've been working me and helping me in the lab. And the funding agency, of course, the Marie Curie INTERCROSSIng and the NPZ linking. So these are some of the references I took. And thank you very much.